Well, good day to everyone joining us and welcome to today's Xtalks webinar. Today's talk is entitled Immune Cell-Mediated Cytotoxicity, Single-Cell Insights Using Droplets and Image Cytometry. My name is Ryan Muse and I'll be your Xtalks host for today. Today's webinar will run for approximately 60 minutes and this presentation includes a Q&A session with our speaker. Now, the webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit your questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Samplix, who developed the content for this presentation. Samplix APS supports the life sciences and medical research communities with microfluidics-based solutions designed to deliver high-resolution insights into cells and genomes. Samplix stands ready to help academic, corporate, and government researchers reach their goals in areas as diverse as engineered cell therapy, molecular engineering, and cell biology. Now I would like to introduce our speaker for today's event. Dr. Sissel Alsing has a background in gene therapy research. She is currently a field application scientist at Samplix, where she supports customers and collaborators in using Xdrop for their single-cell functional assays and integrating the system with existing platforms for analyses. But now, without further ado, I'd like to go ahead then and hand things over to our speaker, Dr. Sissel Alsing. You may begin when you're ready.
Excellent. Thank you very much for that introduction, and hello everyone. I'm looking very much forward to sharing these new results on our single-cell cytotoxicity using image cytometry with you all. But before I do that, I will just introduce Samplix a little bit more in detail. So Samplix was founded back in 2012, and we're headquartered just outside of Copenhagen here in Denmark. But we've also recently opened a small office in the Bay Area of California in the US. Our products include our microfluidics instrument, cartridges, and all the reagents required for cell and DNA encapsulation into our droplets, as well as our online software tools. The outline for today's presentation includes a short introduction to droplet microfluidics, as well as image cytometry, which are some of the methods that we have used to generate the data for this webinar. And then I will talk a little bit more about a multiplex cell-mediated cytotoxicity assay with the single cell readout. So that is the biology which is in these droplets. I'll present the results from the image cytometry and flow cytometry analysis, and then I'll finish by mentioning some of the other applications for which you can use the Xdrop platform, and then summarize key takeaways for today's presentation. Before I continue, I would like to know a little bit more about your experience with some of these technologies though. So we have a short poll question for you.
Yes, so appearing on everyone's screen right now is a polling question, and you can participate by selecting as many of the answers that you see that are applicable and then clicking submit. The question that we're asking is, do you currently use microfluidics, flow cytometry, or image cytometry in your research? Again, you can select more than one answer here that applies to you and click submit, and the answers are microfluidics, flow cytometry, image cytometry, or I use none of the above. Just give you a moment to consider, as the question says, if you use microfluidics, flow cytometry, or image cytometry in your research. Looks like most of you have answered. Thank you so much for your quick participation in this. This looks great. Let's look at your results here. We have 76% of folks selecting for flow cytometry, 24% for saying that you use actually none of the above, and then 12% of you selecting for image cytometry, and then 6% for microfluidics. So thank you very much for participating. I'll hand it back to our speaker.
All right, thank you for that. That's very interesting to know a little bit more about the background for the audience. So I'll mainly focus on the microfluidics and image cytometry part, since also many of you are already familiar with flow cytometry. I'll start by talking a little bit about single cell assays in general though, and why we need single cell assays. So in a heterogeneous cell population, if you run a bulk assay, a bulk functional assay, what you will get is an average profile, which doesn't really reflect all of the individual cells within this heterogeneous population. So this bulk assay masks the heterogeneity and does not allow you to identify any subpopulations that you might be interested in for your downstream work. With a single cell input, such as one that's enabled by our droplet microfluidics platform, you can transform this functional assay into a single cell assay, revealing each cell's distinct profile, and also allowing you to identify the most interesting subpopulations. That could be the most potent cells, the highest secretors, and also to give you a more detailed insight to what is going on in this heterogeneous cell population. Another benefit to using droplet microfluidics is that it allows you to increase the throughput of your assays, going from a very low throughput platform such as doing things in wells or tubes, increasing the throughput in microcytoplates, but with the highest throughput in droplets. At the same time, it allows the user to reduce the assay time, the reagent volume, even the cost of some of their assays because of the reduction in reagent volumes. So this is our droplet generator. Our droplet generator is called the Xdrop, and it produces droplets in a completely automated fashion using these cartridges such as the one you see here on the right side. It's very easy to use from a user point of view. Only a few pipetting steps are required, so no microfluidics expertise at all is required to produce droplets with our instrument. What happens inside the instrument, this automated droplet production, is shown in the video here, which should be running from left to right. So the sample enters here, so that's usually cells in any assay chemistry in an aqueous solution, which enters here at the left side of the video. Then at this first junction, it meets the oil phase, producing these water in oil droplets, so single emulsions. And then at the second junction here, it encounters the outer phase, which is also aqueous. So the result of all this is our double emulsion droplets. So we have the inside here, which contains the cells and any assay chemistry. Then we have the oil shell here, which is surrounded by an aqueous solution, which is usually growth mediums that would be the same as what's in the inside of the droplet without any assay chemistry. So because these are water in oil and water droplets, they are compatible with conventional flow cytometers and cell sorters. And the cells and its secretome and any large molecules are contained inside these droplets, while the thin oil shell allows passage of small molecules, such as gases, which means that the cells can survive for a prolonged period of time inside the droplets. A typical single cell workflow using double emulsion droplets could look something like this. So we typically have an outer buffer and the oil, which are required to produce the droplets. And then we have the reaction mix, which consists of the cells for the assay, as well as any assay chemistry that you'd be interested in. You then produce droplets using the Xdrop. And having produced the droplets, you can handle them very much like you would handle cells. So you can incubate them using a conventional cell incubator. And then they're compatible with different forms of downstream analysis. We commonly use flow cytometry to analyze the droplets. We use conventional flow cytometers and cell sorters. But for this particular webinar, I'm going to focus on image cytometry and imaging, which is a bit of a newer addition to the analyses that we can do with our droplets. For the analysis that we've done in this webinar, we have used Chemometric's Xcyto5 image cytometer, which allows full integration of acquisition, image analysis, and data presentation on the same instrument with the connected software. It has a very high sensitivity, which is on par with high-end flow cytometers, and it's a very flexible instrument in that it allows the user to analyze both adherent cells and cells in suspension, as well as droplets, as you'll see in some of the following slides. There's a lot of different options to design assays. There's a lot of flexibility with 25 different fluorescent channels, 9 different emission filters. And I also, from a user point of view, really like this instrument. It's very easy to use. And the software in particular allows you to plot your data just like you would with flow cytometry data, but then the software connects it to the images that have been acquired using the Xcyto5. The assay that we will be using for this in this webinar is our in-droplet single cell killing assay, where we co-encapsulate two different cell types. We like to use an NK cell line as an effector cell and a K562 cell as a target cell. But you could imagine this being done with different cell types, CAR T cells, and any target cell that they might be specific to. We then co-encapsulate these two different cell types, which have been stained in each different color. The NK cells in blue in this case, the K562 cells with a yellow stain, do the co-encapsulation. And the result will be droplets containing either both of the cell types or one of each. And there will also be some empty droplets in the production. This is because the encapsulation follows a double Poisson distribution, and maybe the most interesting population will be the one containing both cell types, but I also find it very useful that the controls, which would be the background signal from the empty droplets and also the droplets containing just a single cell type, they're already included in the same assay. For this assay, we use an apoptosis stain, which is a caspase 3,7 activated dye, which labels the apoptotic cells. And we do the readout by flow cytometry and image cytometry in parallel. We stimulate some of the samples using IL-15 24 hours prior to encapsulation, while we leave another control sample non-stimulated. For this particular assay, we use an incubation time of approximately 3 hours at 37 degrees Celsius. But as I'll show you later in my presentation, there's probably some flexibility. You can do longer and shorter analysis time depending on the assay. These are just some images from the Xdrop imaging system where you can see these are the droplets. And I've identified here one droplet containing one NK cell stained blue, a K562 cell, which is stained in yellow. And at the same time, we can also see the caspase stain, which is the red one. Here it co-localizes with the K562 cell. So there's an indication that the NK cell, which has been co-encapsulated with the K562 cell, has some killing activity in this droplet. We've actually expanded a little bit on this assay by adding in a fluorogenic granzyme B substrate, which is what is shown here on the right side of the screen. So this is a peptide substrate with a fluorophore quencher, which when it is cleaved, fluoresces in the FITC GFP green spectrum. This is added along with the cells and the caspase dye in the droplets so that we can see which droplets, in which droplet there's active granzyme B release. So this is also an image from the Xdrop imaging platform analyzing the droplets. But taking a little bit closer look at the data that we actually collect from the Xdrop, we get both the bright field image, which you can see here, where we can identify the droplets and also get an estimate of how many cells are inside the droplets. And then we also get the fluorescent signals, of course, the blue one from the NK cell, yellow for the K562 target cell, green for the granzyme B substrate when it's cleaved, and red for the caspase 3,7 dye when it's cleaved. So our collaborators at Chemometric have created an algorithm which recognizes the droplets. And as you can see here, that's the green outline on the right side of the screen. So this algorithm can recognize the droplets based on their shape and define each of these as a single compartment or a single event. What we've done in this first analysis here is very similar to what we would do on flow cytometry. So we have defined each individual droplet as an event, and then for each individual droplet, we've looked at the intensity of each of the signals, the blue, the yellow, the green, and the red. And the analysis that we do with these data is first we like to plot the droplets on a plot such as this with the violet or blue NK cell stain on the x-axis and the yellow K562 cell stain on the y-axis, which allows us to divide the droplet population into 4 different subpopulations. There's of course going to be the empty droplets, which contain neither the K562 nor the NK cells. Then we have the droplet populations containing just a single cell type, that is either the K562 or the NK cell. And then we have the maybe the most interesting population containing both the NK cells and the K562 cells. We can do that both with the stimulated sample and the non-stimulated control. These are very similar. But then we can also gate out this sample with the co-encapsulated cells inside the droplets and take a little bit closer look at the activity inside the droplets. So what we do here is we plot the granzyme B signal on the x-axis and the red caspase 3 and 7 signal on the y-axis. And here we see that there is a lot more activity, both in terms of granzyme B secretion, but also there are more of the apoptotic cells in the stimulated sample compared with the non-stimulated sample. We can use this gating to further divide this population into 4 subpopulations. So there will be, of course, some droplets with no activation, neither granzyme B nor caspase activity. And then there will be some that have apoptosis only, so they're only positive for the red caspase signal, some that only have the granzyme B, and then some where we have both the granzyme B and the apoptotic signal. And I think it's very nice to see that the ones that have granzyme B are generally a lot higher in their apoptotic signal. I think that makes a lot of sense biologically, since the granzyme B is a known cytotoxic mechanism of the NK cells. We run these samples in parallel on a conventional flow cytometer, and we get some very similar results. They look slightly different, but they are quite similar when you gate out the different populations. So here we also first gate out the different populations of droplets containing just one cell type or multiple cell types, and we do the same analysis where we look at the granzyme B secretion and the caspase 3 and 7 signal. The results look very similar, and we can look a little bit more quantitatively at this as well. And that confirms that these methods of analysis are indeed very similar. So in this IL-15 stimulated sample, we see approximately 30% of the droplets that are positive for both granzyme B and apoptosis, and approximately 10% that are positive for either apoptosis alone or granzyme B secretion alone. The non-stimulated controls also look very similar. So this is very nice to see that the results are comparable between the flow and the image cytometry. And that's already a very powerful assay, but with the imaging cytometry, we can dig a little bit deeper into the data, analyzing not only the droplets as one compartment, but analyzing the individual cells within the droplets. So this is another algorithm that has been developed at Chemometic, where we recognize the cells based on their fluorescent signal. And I should say this is a preliminary algorithm. We are working on optimizing the cell recognition inside the droplets. So that's maybe why some of them are slightly, some of them look better than others. But I think we already now get some very useful information about the number of cells, cell types, and also importantly, the co-localization with the caspase signal. This is what you can see here. So this is the cells that have been identified. These are the K562 cells. The blue-orange are the NK cells, and then we have the co-localization with the caspase 3-7. So we can not only see whether there is an apoptotic cell inside the droplets, we can also see which cell is apoptotic inside the droplet. To analyze these data a little bit in more detail, I would like to introduce a new parameter, which is the circularity. And this parameter looks at the shape of the compartment that has been defined as a cell. So in this case, we're looking at the circularity of the K562 cells, which you can see here on the lower image. And then you can, on this side, we look at the circularity of the NK cells, which you can see here on the left side of the screen. And the first thing we do in this analysis is that we remove the droplets that contain only one cell type. So that would be the ones that have a circularity score of 0 for either of the cell types. That would mean none of them are, that would mean there's no cells of this type within the droplet. And that allows us to gate out the co-encapsulated cells within the droplets. We can stratify this a bit more using the same parameter, the circularity parameter, where we can see that some of the droplets they have a very high circularity score for both the K562 cells and the NK cells, signifying that they contain just a single NK cell and a single K562 cells. The ones that have a high circularity score for the NK cells, but a little bit lower circularity score for the K562, they contain just a single NK cell and 2 or more of the K562 cells. The ones that have a high circularity score for the K562, but lower for the NK cells, contain a single K562 cell and 2 or more NK cells. And lastly, there's a population which contains 2 or more of each cell type. And this is some example, some very nice gating from the software where you can recognize the individual cells. And we can use that to have a close look at the activity within these different subpopulations. And what we can see here is that, perhaps unsurprisingly, but that's very nice to see, that is what we would expect, there is more activity in the droplets containing more NK cells compared with the ones containing just a single NK cell. So in the droplets containing multiple NK cells, we see that more than 50% of the droplets, so now we're looking at the granzyme B droplet intensity within the droplet, and more than 50% of the droplets which contain more than 2 NK cells have a positive granzyme B signal. And that's also the ones that contain more K562 cells. When we look at the populations that contain just a single NK cell, it's less than 50% that have the positive granzyme B signal, no matter if there is one or multiple K562 cells that are co-encapsulated with the droplets. We can then continue to do analysis which is very similar to the one that I showed you earlier, with the exception that we're again looking at the droplet intensity for the granzyme B, but instead of looking at the apoptosis signal in the entire droplet, we are looking at the apoptosis signal on the K562 cells only. So whether the compartment defined as a K562 cell is positive also for the apoptosis signal. The results look very similar to what we saw before, that there is more activity, of course, in the droplets containing multiple NK cells and 1 or more of the K562 target cells, with a little bit less activity, but still a significant amount of activity because it is the activated sample. And so there's still some activity even if there's only a single NK cell. So we can look at this again a little bit more quantitatively. And here we very clearly see the increased activity in the droplets containing multiple NK cells, a little bit less activity in the droplets containing just one NK cell. And we can also compare it with the non-stimulated sample, which serves as a control for this assay. And perhaps unsurprisingly, there is very little activity in all of these 4 populations. There is a little bit of granzyme B release in the droplets containing 2 NK cells, but yeah, and a little bit less in the ones containing just a single NK cell. So this is a fairly new application, the image cytometry analysis of the droplets. We have a little bit more data on a flow cytometry analysis, but I think a lot of this could be done in image cytometer as well. So I'll just share some of the data from flow cytometry analysis with you as well. This is very much the same assay where we have co-encapsulated NK and K562 cells in 2 different colors. And instead of the apoptosis signal, we use a PI stain to identify the dead cells. And here we have followed the droplets for encapsulation up to 24 hours, where we see very high increase in the cell-filling activity in the stimulated sample, and also a little bit of activity in the non-stimulated sample. Here we can also take a closer look at the activity in the droplets containing just an NK cell and the K562 cell to get a better picture of the viability of the cells after encapsulation inside the droplets. So the NK cells seem to not really be affected by the encapsulation. There's very good viability for all 24 hours. And for the K562 cells, there's only a small loss of viability after 24 hours of encapsulation. So 24 hours does not seem to be the limit. That's the data that we have for now, but it certainly doesn't seem to be the limit for encapsulation. We also have results, again, this is from flow cytometry, but on the combined granzyme B and cell killing assay. So again, very similar to the assay that I showed you before, the co-encapsulated NK and K562 cells, where we then plot the granzyme B signal on the x-axis and the PI signal on the population with the co-encapsulated cells. And here we can do the analysis as early as 30 minutes after encapsulation. So it's possible to get results very early after encapsulation, and then we follow them for up to 4 hours. And here I think it's very nice that we see a development of the signal first with the granzyme B, which is then followed by an increase in the PI signal from the very same droplets that have the positive granzyme B signal. And we can also follow the cell death alone, which stays more or less stable over the time course. And we can then quantify the granzyme B signal alone and the granzyme B in cell death. So I hope that this has shown you that the Xdrop and our droplets can be integrated into existing analysis platform. We have a lot of data on flow cytometry, which is a nice and well-established platform with a very high throughput. But I think these new image cytometry data also shows you that you can get a very high throughput using image cytometry, as well as a more detailed analysis of what is going on inside the droplets on the single cell level. Of course, it's also possible to do classic microscopy of our droplets, which can get you more detailed single cell analysis, and which is very accessible to a lot of labs, but which of course has a much lower throughput than these other technologies here. In addition to the cell lines that we like to use for our assays, we have also recently published the preprint where we have shown the same assays, a cytokine secretion assay and also a cell-cell interaction assay using these clinically relevant cell types, TILs and CAR T cells, which I think is very interesting. So I highly recommend that you take a look at this preprint. In addition to our single cell functional assays, we also have many different areas of applications for our products, including for genomics workflows, such as for validation of CRISPR edits or to amplify very small amounts of DNA, for example, for diagnosis of tuberculosis and to investigate evolution. Broadly, our applications can be divided into these 3 branches, where we have the cell-cell interaction assays, which I've focused on today. Of course, our cell-killing assay, which can be done alone or in combination with the granzyme B secretion assay, even a cytokine release assay. We can also do secreted protein analysis on the single cell level, where we focus mainly on cytokines and antibodies. And lastly, we have this branch of genomic assays, where we can use our droplets for CRISPR edit validation and insert mapping. So the key takeaways from today's webinar is that double-emulsion droplets enable single-cell functional assays, which can be used on a wide range of platforms, such as conventional flow cytometers, but also imaging cytometers, such as the Excitofly from Chemometic. And using double-emulsion droplets, cell-cell interaction and secreted proteins can be studied at the single-cell level, either in the same assay or as separate assays. The result can be achieved as early as 30 minutes after encapsulation, as shown in the granzyme B secretion assay, but the droplets are stable for much longer incubation. Usually the biology within the droplets will be the limiting factor, since the droplets in the cell, or if stored correctly, can be stable for up to months after production. Lastly, the Cyto5 image cytometer is compatible with Xdrop double-emulsion droplets and also unlocks possibilities for single-cell analyses. That is the end of today's presentation for me. I'd like to thank you all for your attention, of course, and I'd be happy to take any questions that you may have.
Well, thank you very much for this very insightful presentation. I'd like to invite our audience to keep sending their questions or comments right now for the Q&A portion of this webinar, but also direct them to the handouts module where they'll find some additional documentation related to today's presentation to download for some further reading. I've already received some questions though from the audience, so we'll get ourselves started with those. But the very first question I have for you is wondering, will you be able to sell Cyto5 with your instrument?
That's an excellent question. I think we'll have to talk to our collaborators at Chemometric about that to see how far they are with the launch of their instrument. It will be very interesting indeed.
Excellent, thank you very much. The next question is curious, are all flow cytometers suitable for droplet analysis?
Yeah, so we haven't tested them all. There's many flow cytometers available, but in general, our droplets should be compatible with most conventional flow cytometers. We have very good experience with the Agilent Quantion, which we have in-house, and the Sony cell sorter systems. But we also have good experience with the collaborators that have different instruments and customers that also use different instruments. For some of the instrument, it can be a benefit if a 130 micron nozzle or chip is available, but other than that, we haven't experienced a lot of limitations.
Perfect, thank you so much. The next question I have here is wondering, what is the size of the double emulsion droplets?
That's an excellent question. I think I forgot to mention that. So our double emulsion droplets, the ones that we use for our single cell encapsulations, are 60 microns in diameter. That's the inner diameter, which gives them a volume of 100 picoliters. For our genomics assays and our other genomics workflows, we have other droplet sizes available, but yeah, the D50, as they're called, are the ones that we use for our single cell assays.
Perfect, thank you. Now the next question here is curious, what types of cells can be encapsulated with the Xdrop?
Another good question. So as I've shown you today, we have encapsulated many different cell types. We use the NK cells very much, K562 cells, but we've worked with a range of cell types in our own labs. We've even used PBMCs and the TILs and CAR-Ts and their relevant target cells. So as long as it's a conventional size for a mammalian cell and has a kind of conventional morphology, I think it should be possible. For smaller cell types, like I mentioned, we have other droplet sizes available, which might be more suitable, for example, for microbial cells. Yes.
Thank you. The next question's multiple parts. I'm going to break it down a little bit for you. The first is just wondering, how long do droplets containing two different cell types two different cell types inside stay stable? And how many droplets would you expect to get damaged, or what percentage is lost, say, after 24 hours?
That's a good question. So in general, the droplets are very stable, even with multiple cells inside. I think the main limitation would be that from a biology point of view, whether if you overload the droplets a little bit, that could have an effect of the viability. We don't see that with just a few droplets inside the cells, but of course it depends on how many you like. So I guess bottom line is the droplets are very stable for 24 hours, 48 hours, as long as you'd like to incubate, really.
Wonderful. And then as a follow-up, they're also wondering, in co-cultures with two cells, which percentage of the droplets will contain 1x cell A or 1x cell B?
Another great question. So that all depends on the Poisson distribution, and I'm not sure I can do that, off the top of my head. What we usually do is that we aim to get a single cell of our effector cell inside the droplets. That gives us approximately 250,000 droplets containing just a single effector cell out of the 750,000 that we produce in each production. And then we overload the cells just slightly with the target cells because they mainly act as a passive target for the the effector cell. So whether there's one target cell or two target cells, as you also saw in the presentation, it doesn't really change the activity of the NK cell so much. So in that way, only the distribution of the NK cell becomes a limiting factor. But yeah, it's possible to do the calculation using the Poisson distribution, and we have a tool available on our website which can help you do that to kind of get an estimate.
Wonderful. Thank you very much for that answer and for all of the answers, but we have reached the end of our time here today. If we couldn't attend to your questions, know that the team at Samplix will follow up with you, or if you have further questions, you can direct them to the email address on the screen, or of course, visit samplix.com. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. Now, please join me once more in thanking our speaker for her wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank you all. Bye.